So water metabolism. And, or the thing I should remind you first are the fluid compartments. You call them fluid compartments. And those are different compartments, let's say, sealed by different types of, totally different types of so-called membranes. We could call them membrane. One is not so-called, but the other one, or barriers, it's just a very weak barrier. And what are the main two compartments we divide the water in? One is, tell me.
Intracellular.
Very good. So one is intracellular and other one is extracellular. Okay. So this is the main division. That's the crucial one. And over here, if you get an infusion of a pure water, theoretically, so pure water, like really hypotonic water, then theoretically and also very likely it is true, the water also should divide in the same ratio as it is. So normally intracellular, how many, let's say, how does the water divide? Extracellular, intracellular. So 2/3 of the all volume of the water gets or is in the cells. Yeah. So 2/3 are in the cell, and 1/3 stays extracellular, so out of the cells. Okay, and if you get an infusion of free water, let's say, which is not a good idea, but theoretically, then it should divide in the same way. Okay, good. But if you get an infusion of isotonic liquid, that means the liquid has same osmolarity as the water, like everywhere. That means equal to extracellular or intracellular, doesn't matter. Will there be a shift into the cells? No. Okay, it will stay only extracellular because there is no tonicity between these two liquids or spaces. Okay, you won't change the tonicity. So you're just, if it's isotonic, a liquid has the same osmolarity as the extracellular space, and you're putting it intravenously, the liquid will just stay extracellularly. So you will only expand the extracellular space or volume. Okay? Yeah? That's very important to understand. And then extracellular space still divides into what? Into intravascular space and interstitial. Very good. Okay, so I'll put the IC still here, that we have it next to each other. Okay, and the boundaries are natural, and this is what? This is cell membrane. Okay, so this is a semipermeable membrane that divides the extracellular and intracellular space, and then there is sort of a membrane. It's sort of a joke in a way, and that's the capillary membrane. Okay, watch out, not arterial. Arterial wall is thick and nothing goes through. Okay, it's a pipe, but it's not fenestrated. But capillaries are. So sorry, this is so capillary wall. Okay, and you already learned and you are gonna hear it for the second time, I guess, about some substances or that are passing yes or not through these, and we're picking up the most important one. Of course, we don't care about other ions. We care mostly about sodium, for example, and the other ions behave the same. Okay, so first, if we start with water, is water able to go through the capillary wall? Very easily. Okay, is water able to get into the cells? Yes. Through what? Now we found out there are aquaporins actually in all the cell membrane. So finally we understand why water, which is water, gets through a lipid bilayer because they're aqua points actually. Okay, fine. Then what about urea? Urea. It's a special molecule. And it's very good that we're mentioning it here, because thanks to urea you will understand many other principles, especially when I'll be explaining tonicity very soon. So urea behaves very likely because of behaves like water in a way. So it goes very easily through the capillary wall and very easily passes through the cell membrane. Okay. And now sodium. How is it with sodium? Sodium.
That's the question.
Yeah.
The urea, how does it go through the intracellular?
Very likely, I think it goes with the water through the channels. Okay. Yeah. I think so.
Okay. Through the aquaporins.
But hey, I don't know the molecular details, and it's not important for pathophysiology. It just gets to the cell very easily. Okay. And I suppose it uses the aquaporins as well. It's everything about the polarity of the urea, although it's a molecule. It very easily passes almost all the walls in the body, except in kidneys. That's a special thing. Yeah, in kidneys it's made a bit different. Okay, but once urea goes up, it gets everywhere. It gets into your brain. It gets into your GIT tract. Unfortunately, in GIT tract, it crystallizes, and it forces you to vomit and have diarrhea. So it makes pretty nasty things, urea. Okay, and it's big. And also, of course, if the patient is not on dialysis, when urea goes up, it gets on the skin and you stink. You stink like urine thanks to urea. Okay, yeah. So you can smell the patients. Okay, and that happened before we had like dialysis for everyone. Okay. So like 30, 40 years ago, when only young people could get dialysis, the older ones died and they were stinking by urea. Okay? Okay. So, but anyways, urea passes easily, but in contrast to that, sodium gets easily into the interstitial space, but needs a NAK pump to get into the cells. That's crucial. And then you have glucose. And glucose is a very important molecule, very important and very osmotically active. Okay, of course, sodium is the big guy in terms of attracting water. Sodium is the very powerful, but glucose as well. It's pretty powerful. So, and glucose can penetrate easily into the interstitium, but not into the cells. And now, last ones is albumin or proteins in general, but albumin is the smallest one. You know, albumin is responsible for oncotic pressure, and normally it should stay in the vessels. But in case you have inflammation locally, albumin gets into the interstitium and makes edema over there. Okay? Yep. Or if you have a stroke and the blood-brain barrier gets damaged, and also you have escape of albumin, and then you have edema over there. So the albumin escapes the capillaries. Okay? Yeah. So, but normally, albumin should stay inside the vessel. Okay? So that's just to recap. But what is the formula for osmolarity? What is it?
Glucose plus sodium.
Very good.
And yeah, times 2, yes.
Plus concentration of
urea.
Very good. And urea. First of all, there's no logic in my colors. I just want to make it colorful, okay? So don't expect any. I'll sometimes try to keep the colors, but yeah, no chance. So this is the formula, you know. But is this exact? No. If you want to exactly know what is the osmolarity of the plasma or of the blood, you should add all the substances or the particles that are in the blood, and you should add them together, and that would be exact. Okay? But basically we don't need to do that, because we are with this formula very near the truth. Okay, and why is that? Well, if you know these ion columns, I would call them ion columns. And
The anion gap columns.
yeah, yep. So over here you have a cations and anions, and the important thing is that there should be equilibrium between cations and anions always. Okay, so normally if I would want exactly to know the osmolarity, I should count everything together, but I can cheat because I know the rule of electron neutrality. Then, and I know the highest amount of cation that is of sodium. That's 140, isn't it? 140. And that's the main cation. And the rest of them, what is that? Potassium, that's only 5 or 3.5. Or then there is calcium, that's only 2 millimoles. Then there is magnesium. Okay, so calcium, magnesium. And basically you can forget these. Because what is that compared to 140 of sodium? You can ignore those. Okay, and then what's the main main anion, which is chloride?
Chlorine.
Yeah, so it's chloride. There's a bit less of chloride than sodium.
100.
Okay, yes, and then you have still what? Bicarb. Okay, that's 24 about. And then you have A gap because there are other things over there like albumin again and lactate if it's built up and whatever. So there's the rest of the anions. You call it A gap, anion gap. Okay. So if you look at this, okay, you could either add all of the ions and cations together, or you can cheat, and you can just take sodium, measure sodium. Basically, if you multiply it by 2, you know it has to be equilibrium, so you get the same amount of anions, and you can forget this because this should be like in reality. If I would draw it in a ratio, it would be really small, not as big as over here. Okay, so basically, look at this. Maybe I can forget glucose and urea as well, because what is the normal glucose level? What is it? It's
3.5.
3.6.
yeah, 3.5 or till 5.5. That's nothing. It's like potassium, so I can forget it as well. And what about urea? It's 3 point. Yeah.
7.5 to
Yeah, or
7.
it always depends. Depends what tables you're using. But I like 3.8 till 8.3 because it's just a backwards number. Okay. So these are the amounts. So basically, yeah, do you agree that I can ignore urea and glucose? Yep, I can ignore that.
Another question. Like,
Yep.
this is the rate. This is the amount of glucose before eating. After eating, it can become 14.
No,
or even
no, never over 14, never over 10 in healthy
I think
person. Okay,
you're
never.
asking about the
Watch out. Wait, still. You'll get the answer. So why am I still... We could really, in healthy person, and now you have the answer, we can ignore also glucose and urea. But we are not ignoring it because of 2 crucial diseases. Okay? And one of them is diabetes. And the other one is kidney failure. Okay? So kidney failure. And these ions can really go high in a disease. And you can have 80 millimoles of glucose in case you are decompensated with diabetes. It's really high. And you can have really high urea as well when failing with kidneys. So that's why it's stressing the formula. You shouldn't forget never to check the glucose and urea, because although if you would measure only osmolarity, basically you can never be sure that there's nothing really wrong, because the number could be normal, but the person could be in a serious problem. Okay, yeah. It's pretty the same idea like with pH. You can have a normal pH, but you can be really in trouble because you have a combination of, I don't know, respiratory alkalosis and metabolic acidosis. Okay, yeah. So basically, number which talks about osmolarity can tell you there's something wrong when it is way out of range. When you have like 250 osmolarity or 330, then you know there's something wrong. But you can be never like sure that although you're having 280 milliosmoles, that the person is fine. Okay, and now we're coming to the semipermeable membrane, or in other words, in our case, cell membrane. And if I'll draw the cell membrane over here again, this is crucial. If the glucose goes up, and watch out still, remember, if you are eating whatever, sugar, etc., and glucose gets into your urine, you are very likely having diabetes mellitus. Healthy person who eats whatever sugar, the boost of insulin should keep it below the threshold for kidneys. Okay, so glucose should never go over 10, or some books talk about 11.1. Doesn't matter. Remember 10. Okay, but anyways, in this case, this is a whatever. If glucose goes up, like, doesn't matter. It's just now we're talking about, there's lots of glucose. Glucose is not able to pass the cell membrane easily and fast. That's why the osmolarity will be increased over here. Yep. And if this is extracellular space and this is intracellular, the hyperglycemia is going to suck out the water out of the cells and they will shrink. And this, of course, has an impact on neurons. So that's why in high hyperglycemia, you can really, in few days, as the hyperglycemia in diabetic patients goes up and up and up, they can get into coma, diabetic coma. Okay, yep. And one of the mechanisms of the coma is the tonic effect. Okay, yeah, effect. So, but look at that. If urea increases over here, it will very instantly equalize, get in and equalize over here. So look at that. If we have this specific membrane that is permeable for urea but not for glucose, then in this case, urea will make no shifts of water. There will be no shifts, but glucose will. Okay? Yeah? So it's fully permeable for urea, but not for glucose. That's why also watch out. Because if you have increased urea, like if you have, I don't know, if it goes to 30, and and everything else is gonna be still normal, so sodium is normal, and then let's say you're gonna have 330 milliosmoles. So this sounds like crazy, but in reality, this will make no shifts of water, the increased urea. Okay, so you can sort of be fine with that. Okay, so it won't change it won't change the amount of extracellular water and intracellular water. Okay, but glucose will, increased glucose will. So really, you should really think if you're going to sometimes add the urea into the formula or not. Okay. Because also, what do you think, what about the osmoreceptors in hypothalamus? And you already heard how they work. They work those are normal cells, but they're very sensitive to changes of the volume. So in case I'm having my osmolarity of the blood increases, or of the extracellular space, by glucose, whatever, they shrink. The cell will shrink, the receptor in the hypothalamus, and then sends a signal, and you're going to be thirsty and released a hormone, and that hormone is ADH. Okay, so this is how the osmoreceptors work. So they will react to increased levels of glucose, but they will not react very well or at all to urea. Understand? And now we're coming to this really important term, which is effective osmolarity. And glucose and sodium and all the particles that are not able to get easily into the cell and they need active transport, they are creating effective osmolarity, or in other words, tonicity. Okay? Effective osmolarity. Tonicity. So you can say that sodium and glucose, they have tonicity. They will create tension. They will shift the water, but urea will not. So you don't count urea under tonicity. Okay? Okay? In case we're talking about cell membrane. Okay? Because of course, if there's going to be a membrane with different features, like for example capillary wall, okay, so the only effective tonicity, the only thing which makes effective tonicity in case of these substances is which one? Which one?
Again, please.
If now we're gonna talk about capillary membrane. So which one out of these makes, yeah, this is the only one which will sort of make water to shift, to keep it in the arteries. Okay, get it? So it's always about the features of the let's say substance of the particle and the features of the membrane. Okay, yeah. So these are not tonically active if we talk about capillary membrane, but they're tonically active if we talk about cell membrane. Okay? Yep.
So this is a question. So just if I understood. So effective osmolarity is when the substance contributes or changes the tonicity of the cell.
Yes. Or tonicity, it's the feet we talk about if it changes if it shifts the water somewhere. Okay? Yeah.
Yes. That urea, for example, which is not effective, doesn't contribute for effective osmolarity.
Yes.
It changes osmolarity, for example, in the kidneys, right?
Yeah, in kidneys, it's a different membrane again. Exactly. This one is not permeable for the urea. It's specially made over there. Very good. That's actually I mentioned it a bit when I talked about, I made a small comment over there. But very good. Yeah, exactly. In kidneys, that's a special case. Okay? Yep. It's made that you really need to, you need ADH to bring the aquaporins over there to the collecting duct. Otherwise, the urea won't go through. Okay? So the, let's say the medulla of the kidneys, it's a special place. Okay? Yeah? Okay. But good. But now, so you understand the differences between like total osmolarity and let's say effective osmolarity. Okay? And now the big question. So what happens when urea goes up with ADH secretion? There is no ADH secretion. Yeah. But what happens when glycemia goes up? And what happens to for example, to sodium when glycemia goes up? Tell me. And that's what you typically see.
If glycemia.
Yep.
Could you please repeat the question?
Yeah. So what happens with, for example, sodium concentration when glycemia goes up?
So when glycemia goes up, the ADH will also go up, and that influences the sodium retention and reabsorption in the kidney.
Watch out, watch out, watch out, watch out.
So we'll.
No. Because ADH
No.
plays with water and urea. It's not aldosterone. Watch out.
Yep. Oh yeah, true.
Okay, but you're heading right. That's what I wanted you to understand. That every time the glycemia goes up, you should think of it stimulate, because you're gonna have hyperosmolarity, and this triggers the central osmoreceptors to release ADH. And as a second step, this forces you to drink water and retain water. And that's why you're going to dilute everything else. Also glucose, but you're going to dilute natremia. So basically, if someone has glycemia and increased osmolarity, the body will decrease osmolarity by diluting everything, and you're going to have hyponatremia. Okay? So this glycemia changes your natremia to hyponatremia. Okay? Through ADH secretion. That's why when you check the diabetic patient, at the end he can have, if you add the glucose and everything, he can have a normal osmolarity, because glucose is 60, but sodium is decreased. It's not 140. Okay, yeah, it's gonna be 120, 110. And
Yeah,
this,
that's what I was gonna ask.
very good. And you call this, and watch out, you call this pseudo-hyponatremia, because in reality the absolute number of sodium is fine, and also you won't be doing anything with sodium. And someone has hyponatremia, you won't supplement it with sodium. You will correct the glycemia.
The glycemia.
And the sodium should normalize itself. Okay, good.
Wouldn't sodium be lost in the urine though?
Well, depends. Of course, then other mechanisms will turn on. But you're having hyponatremia, so you won't be getting rid of sodium. Okay. Of course, depends. Then maybe depends how much polyuria will have, because also increased glucose causes polyuria. So then it's gonna get pretty wild in a way. But remember, when someone has hyperglycemia, the other ions will be diluted. You're going to have hypokalemia, hyponatremia, hypocalcemia, okay? And you're going to then correct the glycemia, and the other ions should normalize, okay?
No, I understand. I'm just thinking because for diabetics, right? Diabetics usually, as a consequence of it, the victim would be dehydrated.
Yep.
And sodium would usually follow water, or is it?
No. No, water follows sodium, okay? Water follows sodium. So you can have some losses, and you're right, diabetics are dehydrated, so they're going to be in hyperosmolarity, but the hyperosmolarity is mainly due to the hyperglycemia, okay? Yep. And the sodium is going to be decreased, although, yeah, depends how much you're dehydrated, and then it's going to be increased, okay? Yep.
All right.
Okay, so, and maybe, okay, so this is clear. So this was about the fluid compartments, and as well about the osmolarity and the difference between like total osmolarity and the effective one, okay? And remember, urea is not changing tonicity. So, thanks for watching, and don't forget to subscribe and ring that bell. And as always, check the description below for supplementary questions and other materials.